Good afternoon, learners. I welcome you all again to this Cadherin session. In my last presentation last month, we took up one course that is cell death and renewal. Today we are going to take another unit that is cell junction and extracellular matrix. This is a part of your molecular cell biology course, that is MSCZOO 001, and you will find this unit in block number 4. Now actually there are two components, that is cell junction and extracellular matrix. Today we will be focusing on the cell junctions only. So we will try to understand what is meant by cell junction and what are the different components that form part of the cell junctions. Now to start with, what is meant by cell junction? Now you all know that an eukaryotic cell, it is multicellular. Okay, that means it is composed of more than one cells. Now these cells need to communicate. Okay, and therefore in order to mediate this communication, there should be some sort of cell-cell interaction, or there should be some sort of cell-cell junctions. That is, between two cells, there are some points which are actually meant by the junctions. Now these junctions can be classified into three different groups. One we call as the occluding junction, one anchoring junction, and the third communicating junctions. Now we will try to understand what does it mean. The first one, that is occluding junctions, which are also known as tight junctions. Their main function is to arrest the communication between two cells. I mean, they actually seal the junctions in such a manner so that there is no transfer of any molecules, chemicals, or any kind of components between two cells. So through this type of junction, there is no exchange of materials between two cells. That is occluding junction. Second is anchoring junction. Anchoring means two cells in a multicellular organism, the two cells need to attach, okay? They need to attach, or they need to be held together, as well as each cell should be attached to the cytoskeletal elements, that is the actin, mostly the actin, or to the extracellular matrix. So the cells need to attach between themselves, and the cell also needs to attach to the extracellular matrix. So those junctions, they are known as anchoring junctions. And next, the communicating junction. Communicating junction is just the opposite of the occluding junction or the tight junction. That means in case of communicating junctions, there is exchange of metabolites, exchange of chemicals, exchange of ions between two cells. So communicating junction allow free passage or free exchange of ions between the cells. So occluding junction function is to stop the exchange. Anchoring junction, mainly their function is to mediate the anchorage, and the communicating junction, their function is to allow communication or exchange. So we will now take a detailed look of what these junctions are composed of. What are the structural components that make up these kind of junctions? So you can see again, that is under occluding junction, we have tight junctions, septate junctions. Tight junctions we call it in case of vertebrates, and septate junction we call it in case of invertebrates. Then anchoring junction, as I already told you, that the cell needs to attach to the actin filaments, In that case, we call it cell-cell junction or the adherence junction. And if the cell interacts with the extracellular matrix, we call it focal adhesions. Also, apart from actin, there is other cytoskeletal element, that is intermediate filament. And the cell also needs to connect with the intermediate filaments. So in this case, like desmosomes or hemidesmosomes, the cell will interact not only with the actin but also with the intermediate filaments. And finally, the communicating junction, that is, we call it gap junction, and in case of plants we call it plasmodesmata. So those are included under the communicating junctions. So now we will take each of these junctions one by one, and we will try to see what are the different structural components which actually make up these kind of junctions. So we start with the occluding junction. As I said, that it is also called the tight junction. Now try to understand why these type of tight junctions or occluding junctions are needed. These type of junctions are mostly found in those cases where you do not need this kind of free exchange of proteins. Like, let's imagine an intestinal surface. Now this intestinal surface, it has an apical surface and a basolateral surface. The proteins which are present in the apical surface and the proteins which are present in the basolateral surface, they are very unique. So these type of proteins, the proteins which are present in the apical surface of the intestine, let's say small intestine, they should not come or they should not get translocated to the basolateral surface. Similarly, the proteins which are present in the basolateral surface, they should not come in the apical surface. That is, these type of intestinal surfaces, they have a distinct polarity. And there is a directional transport, and that directional transport is actually maintained by this kind of proteins. So in these cases, what do these tight junctions help? How do the tight junctions help? The tight junctions help by not allowing the protein which is present in the apical surface to come to the basolateral surface, or vice versa. The proteins which are present in basolateral surface should not come to the apical surface. So this kind of regulation or this kind of restriction is maintained by the tight junctions. So these tight junctions, they mostly help in the directional transport, and they are mostly present in those kind of cell surfaces where there is a polarity, where there is a distinct apical surface and a basolateral surface. So they block this kind of diffusion, that is, from one surface to the other, the protein components should not get translocated or diffused, and that will help to maintain this directional transport. So the tight junctions between epithelial cells, they act as barrier to the diffusion of some membrane proteins and lipids between apical and basolateral domain of the plasma membrane. They seal the neighboring cells. Now how can you experimentally show that the tight junctions, they arrest? If you see, if you add any tracer molecule to one side of epithelial cell, so it can be intestinal cell or it can be an epithelial cell also. So if you apply a tracer molecule and add it to one side of the epithelial cell, you will see that it will not pass beyond the tight junction. So you can see that in the upper part you are getting the tracer signal, but in the lower part you are not getting the signal, because in between you have the tight junction, and therefore you can experimentally show that there is no exchange across the tight junction. So this proves that the tight junction, they arrest the translocation or they arrest the diffusion. So this has been experimentally shown through the addition of the tracer molecule. Now, tight junction lining the small intestine are 10,000 times more permeable to inorganic ions such as sodium than the tight junction in the epithelium lining the urinary bladder. So, there is a distinct difference in the property of the tight junction in different cells. That is, in the intestinal cell and in the epithelial cell, their properties are different. These differences reflect differences in tight junction proteins that form the junctions. Now, in this particular figure in the right hand side, it shows that what these tight junctions are actually made up of. The tight junctions are actually made up of a huge network of an anastomosing strand, anastomosing network of strands. And these strands, which are marked in green in the right hand side diagram, they encircle the apical end of each cell in the epithelial sheet. So, the ability of the tight junction is to restrict the ionic passage. So, basically what they are composed of? They are composed of some thread-like structures, anastomosing thread-like structures, and a careful examination of this network, that is strands. I will come to the next slide. There you will see that it is composed of a series of protein. So, the next slide shows that how these tight junctions or how these strands, they seal together the cells. So, you can see in the left hand side figure, you can see that you see the two cell surfaces, and the two cell surfaces are actually tightly sealed by some chain-like structures. See the marked with green. So, as like you fasten something with a chain. So, similarly here the two surfaces of the cells, as if they are fastened together or tightened together by some chain-like structures which are marked in green. So, this is how the tight junctions actually they are organized or they are arranged, and thereby they seal the cells. In the right hand side figure, you can see that the cell 1 and cell 2, it's given the two surfaces of the cell, and in between those marked with green, those are actually the tight junctions, and the diagrammatic representation of the tight junction, and it shows that there are some 2 or 3 proteins that make up these tight junctions. Those proteins, mostly the most important protein is called the claudin, and another protein that you have is called the occluding. So, claudin and occluding are the two tight junction proteins. Now, we will take a look at, so now this claudin, this is a major transmembrane protein. What is meant by transmembrane? Transmembrane means which spans across the membrane. Transmembrane protein, and it is the major protein, major tight junction protein, that is claudin. Obviously, if somebody asks you what is the function of claudin, your answer will be that because it is a tight junction protein, so its primary task or its primary function will be to seal the cells. That is to seal the apical and the basolateral surface of any cell. It can be epithelial cell or it can be, as I said, intestinal cell. But apart from that, these type of claudins also have some other functions. So, what is that function? You must have studied about the tubular reabsorption in case of kidney, tubular reabsorption in case of a nephron of a kidney. That is, when the urine is eliminated from our body, the waste water in the form of urine or excess water in the form of urine is eliminated from our body. Then before elimination, what happens? In the tubules, what happens? There is a tubular reabsorption. That is, the essential ions like magnesium, they are being reabsorbed before the waste water in the form of urine is eliminated. So this is called as tubular reabsorption. So this kind of Claudin protein also helps in tubular reabsorption. That is, the magnesium ion is being reabsorbed from the urine into the blood before the urine is being eliminated. So this is a very, very vital function of the Claudin. If there is a mutation in the Claudin gene, then what will happen? Then it will result in excessive loss of magnesium from the body. So it will be a wastage. So excess magnesium, because magnesium is required for so many vital functions in our body. So the magnesium is eliminated from the body. If the Claudin gene is mutated or if the Claudin protein is non-functional. So that is another very important function of Claudin, which is a tight junction protein. Second protein, that is also a transmembrane protein, that is called Occluding. But the very detailed function of Occluding is not very clear. It helps in the tight junction, it helps as a component of tight junction, but other than that, whether it has any other major physiological function like the Claudin, that is not very clear to us. The third protein, which is called ZO protein. ZO stands for zonula occludens. So this is another protein, and there is an interaction of the Claudin and Occludin with the ZO proteins. And their function is almost the same, that is, they anchored the strands to the actin cytoskeleton. So apart from these three proteins, there are several other proteins also, but details of them we are not discussing here. But since these three proteins are the major proteins, so we have mentioned their names and their precise functions. Now, as I said, in case of invertebrates, we do not call them as tight junction. There we use the term septate junctions. For example, in Drosophila, it is an, it's an insect and it's an invertebrate. Here, people have actually studied or people have examined the junctions, and they have named it as the septate junctions. The organization of the septate junction is a little bit different in case of invertebrates. Morphology is very peculiar or distinct, because here, as you can see in the figure, that it is a continuous band-like structure. As it's a, you can say, a sort of a ribbon-like structure. Okay, and there is the proteins, they are arranged in parallel rows with a regular periodicity. So the morphology is not like you find it in case of vertebrates. In Drosophila, then what are the proteins that make up this that make up these septate junctions? There is one protein which is called discs large. Like there you have studied about Claudin, Occludin, ZO protein. Here the protein which is present is known as discs large protein, and that protein forms the septate junctions in Drosophila. And this protein, it is structurally related to the ZO proteins. Now what will happen if this protein is mutated? If Drosophila, they have the mutated that is the discs large protein, what will be the consequence? The consequence will be, number one, the septate junctions will not form. That is very clear. Second point is, they develop, these kind of flies, they develop epithelial tumors. So what does it prove? It proves that this kind of discs large protein not only plays a structural role, but they also regulate cell proliferation in the epithelial tissues. That means they also take part in the cell signaling process. That is the relay of the intracellular signals. And these signals, they actually originate from the occluding junctions. So if these proteins are mutated, then there is a tumor formation in the epithelial cell layer. So overall, what does it prove? Overall, it shows that the tight junction or the septate junction, they not only play a structural role, but they also help in maintaining the cell signaling process and also some basic physiological processes, the basic animal physiological processes. Okay? So very, very vital molecules. The junctions are very important, and the proteins that make up these junctions are important not only from the structural point of view, but also from the physiological or the cell signaling point of view. So that is all about the tight junctions. Now we come to the second category, that is the communicating junctions. Now communicating junctions, as I said, these are also known as gap junctions. What is the difference between tight junction and gap junction or communicating junction? The difference is, as I told you, gap junctions, they will allow the exchange. So if you imagine two adjacent cells, and obviously there are the cell membrane, and these two cells are separated by a very narrow gap of about 2 to 4 nanometer. But although there is a gap, there is exchange of molecules, because that gap is actually spanned by some proteins, some channel forming protein. And these channels will allow inorganic ions and water soluble molecules to pass from the cytoplasm of one cell to the cytoplasm of the adjacent cell. How will you show it experimentally? Again, if you use some tracer molecule, or you can use some fluorescent dye, and you tag certain molecules with that fluorescent dye in one cell, but you will get the fluorescent signal in the adjacent cell. Why? Because there is exchange. And therefore, you get the signal, fluorescent signal, not only in one cell, but also in the adjacent cell. So there is something in that gap that allows the free passage of molecules from one part of the cell to the other. For example, in the neuronal transmission, the synaptic transmission, where the signal is relayed in the nerves. So there in the synaptic cleft, there is this kind of gap junction, because the signal has to relay from one cell to the other, from one neuronal cell to the other. So in the synaptic transmission, you will very well find this kind of gap junctions. So sharing of the molecules, in short, sharing of small metabolites and ions. Now next we see that what are these gap junctions composed of? Again, these gap junctions are composed of certain proteins. The name of that protein is called connexin, and this connexin is a 4-pass transmembrane protein. That means it spans across the membrane 4 times. And this protein is composed of 6 subunits. Each of these subunit is called connexin. So connexin is the total protein, and it is composed of 6 subunits. Each subunit is termed as connexin. And you can see in the figure that in the diagram you can see that the one in the which is shown in green, that particular diagram, you can see that there are two different kinds of patterns, or rather three different kinds of pattern. One is totally light green, one is light green, deep green combination. What does it mean? It means that the subunits can be of the same type. Like all light green means they are similar type. 6 subunits are identical. So it's you will call it a homotypic. That means single kind. That is they are all similar. But if the subunits are different, then you obviously will call it heterotypic. So the subunits can be of same kind, similar kind, or the subunits can differ. So accordingly, there are two different classification, or there are two different kinds of you can say the connection molecule. Now, they form a continuous channel, aqueous channel. They protrude from each cell surface holding the interacting plasma membrane. It is composed of 6 identical protein subunits called connexin, or it may not be identical also, as we have seen in the diagram. Connexin has a central aqueous pore lined by one transmembrane alpha helix from each subunit. So you all know that the proteins, they have different conformation, alpha helix, beta sheet. So basically these are channel proteins. Why channel? Because they allow the passage and no energy is required for that. Like pumps which you have in the membrane, the proton pumps, sodium potassium ATPase, they require energy. But here it is just the diffusion, the channel proteins. These are basically channel proteins. Unlike the pumps, the proton pumps which you have in the membrane. Now, in case of plants, so whatever we discussed, these are mostly in animals. But in plants, these communicating junctions are known as plasmodesmata or plasmodesma. So how do these plasmodesmata look like? Again, you can see in the diagram. Particularly if you look at the right hand side diagram, here you can see the cytoplasmic channels, they pierce the plant cell wall, because in plants not only plasma membrane but you also have the cell wall. Outside the plasma membrane you have the cell wall. So the channels pierce the plant cell wall and connect all the cells in a plant together. And if you see the structure of the plasmodesma, which is singular and plural is plasmodesmata. So if you take each plasmodesma, you will find that it is lined with a plasma membrane that is common to the two connected cells, and there is a fine tubular structure. So you can see that there is a tubule like almost red colored, you can see, or slightly brownish colored tubular structure you can see, which actually penetrates, and that tubular structure we call it as desmotubule, and this desmotubule actually originates from the endoplasmic reticulum, that is the smooth endoplasmic reticulum. You know, cell endoplasmic reticulum is a cell organelle. So it originates from the smooth endoplasmic reticulum and it pierces through the intervening cell walls. So this is a cylindrical channel with 20 to 40 nanometer in diameter. So this is the description. As you can see that through the center of the channel is a narrower cylindrical structure desmotubule which originates from the smooth endoplasmic reticulum. Between the outside of the desmotubule and the inner face of the cylindrical channel, there is an annulus of cytosol through which the small molecules can pass. Plasmodesmata form around elements of smooth endoplasmic reticulum. ER stands for endoplasmic reticulum. and assembled during cytokinesis. Plasmodesmata allow passage of molecules with molecular weight less than about 800. So the main idea or the main take-home lesson is that number one, the protein, that is connexin, the structure of the connexin, function of the connexin, and in case of plant, what is the structure of the plasmodesmata. Okay, and basically it's the desmotubule which actually allows or which actually helps or acting as a channel through which the molecules can pass. And the origin of the desmotubule is from the smooth endoplasmic reticulum. So that is all about the communicating junctions. Now we come to the third kind of cell junction, and this is, we have to study, this is a bit descriptive or this is a bit elaborate, and it's a detailed kind of structure. That is the anchoring junctions. Now as I told you, that anchoring junction is a little bit different from the tight junction and communicating junction, because anchoring means anchorage. It comes from anchorage, that is attachment. So who will attach with whom? There, between two cells there can be an attachment, and as I said, the cell will also attach to the substratum. Substratum means the cytoskeleton, that is actin or intermediate filaments, or the attachment should be also with the extracellular matrix. We will read about extracellular matrix in our later session, in the coming sessions, but today we just introduce you to the term, that is extracellular matrix, which is mostly made up of components like glycosaminoglycans and some proteins like collagen, elastin, fibronectin, laminin, etc. Now, the cell connects with the anchoring junction with the help of certain junctions which are called adherens junctions. Most abundant in tissues subjected to severe mechanical stress like heart muscle and skin epithelium. So as I said, attachment should be with actin, attachment should be with intermediate filaments. For example, with the desmosomes and the hemidesmosomes. Now see this diagram. What does it show? See, there are two cells, cell 1 and cell 2. Now cell 1 is attached to the cell 2 through some components which are marked with green, and that green structure, it has a head portion and a tail portion. The tail portion is, how does it look like? The tail portion is within the cytoplasm and the head portion is outside the cytoplasm. Similarly for the other cell also, the head portion is outside the cytoplasm, tail portion is within the cytoplasm. Now the tail portion, it is attached with the actin, that is marked in red color. Actin filaments are marked in red color, and the tail portion is connected with this red colored, as it is shown in the diagram, red colored, with the actin filaments through some other proteins which are marked in blue. Again, the actin proteins or the actin filaments, they are connected with the extracellular matrix which is marked in yellow, again with certain proteins. Those are again marked in blue. So all these proteins which are present in that blue region, those are all helping in anchorage. So all those proteins they form component of adherence junction or anchoring junctions. So we will focus our attention on that blue component and also on that green component, because green component, how is it helping? Green component is also helping in attaching the two cells. Okay, and that green component, it is a very elaborate structure which is having a head portion and a tail portion. So we will now focus our attention on the green structure and the blue structure. Okay, so intracellular anchor protein, they form a distinct plaque on the cytoplasmic phase and connect the junctional complex to either actin filaments or intermediate filaments. But additionally, there is an extracellular domain also, and therefore the intracellular anchor proteins, they also bind to the extracellular domain, and that interacts with the extracellular matrix or the extracellular domain of specific transmembrane adhesion proteins. So we take a look at that protein. So this is actually that protein. See, if you now focus our attention to that green proteins, that is one having a head portion and a tail portion, then the head portion, you can see that there are, the head portion, it has 5 repeats and it's a dimer. 2 side by side you can see components. So it is a dimer, and the head portion is composed of 5 repeats, and the tail portion, it is within the cytoplasm. Now in the head portion, you can see some red colored spots. Those red colored spots are actually the calcium binding pockets. That is where the calcium ions bind. These proteins are called cadherins. So cadherins are very important component of anchoring junctions. It is through these cadherin proteins that a cell attaches or anchorage with another cell, anchors with another cell. It mediates anchorage. But this is calcium dependent process, because whenever this kind of attachment is required, the calcium ions come and bind to the calcium binding pockets of the head portion of the cadherin repeats, and thereby the molecule gets rigid or stiffened. If the calcium ion is absent, or if you add some component which chelates calcium, like EDTA, ethylene diamine tetraacetic acid, if you use a calcium chelator, that will chelate calcium, that will snatch away calcium, then there is a calcium deficiency. And because of that calcium deficiency, what happens? The cadherin molecules are not rigidified. It becomes a floppy structure, as you can see in the right hand side of the figure, that it's a floppy structure, and because of that floppy structure there is no attachment. But when there is abundance of calcium, then the structure gets rigidified and it mediates communication. So this is why we call it a calcium mediated cell adhesion molecule. We call cadherin as a calcium dependent or calcium mediated cell adhesion molecule, which helps in adhesion between two cells. So that is the structure of a cadherin, and that is how the cadherin molecule functions in presence of calcium. Now, as we have introduced you to the term adherence junction, or we also call the adherence junction as adhesion belt. That is, in this kind of junction there is a continuous adhesion belt which is present below the tight junction, encircling each of the interacting cells in the sheet, epithelial sheet. Now, another point that I want to mention is, if you now take a look at this diagram. So now by this time, all of us, we have learnt about cadherin dimer and how the cadherin molecule gets activated in presence of calcium. Now we focus our attention to the tail portion. Now see the tail portion, that is, it's a kind of, tubular kind of structure, the tail portion, which is within the cytoplasm. See, it is connected with the actin filaments through some proteins which are marked with blue. What are these proteins? Now, there are three different kinds of proteins which are called catenins: alpha catenin, beta catenin, and P120 proteins. So these proteins, they help to connect the cytosolic portion of a cadherin to the actin cytoskeleton, and that is how they mediate interaction or anchorage with the actin filaments or the cytoskeletal filaments. So cadherin, extracellular part of the cadherin is mostly involved in communicating with an adjacent cell or mediating anchorage with the adjacent cell, whereas the cytosolic tail portion is actually responsible for interaction or anchorage with the actin cytoskeleton. So both the head portion and the tail portion are equally important. If you delete the tail portion, what will happen? It will not anchor with the actin. If you delete the head portion, what will happen? It will not mediate anchorage with the adjacent cell. So E-cadherins which lack their cytoplasmic domain, they also cannot hold the cells together. So I hope you understand the structure of the cadherin and how each of these domains or each of these portions, they help in attachment. And the region where these kind of cadherins are located, we call them as the adherens junction, which form a kind of belt-like structure called adhesion belt. Like we wear belt. Okay? Why? To fasten the, better fittings and to fasten. So similarly, it's also a belt-like structure. So catenins, vinculin, alpha-actinin, these are some of the additional intracellular anchor proteins. So that is all about the adherens junction and the role of the cadherins. So it's a calcium dependent cell-cell adhesion, but you have to understand, though we will not focus our attention much on calcium independent adhesion. In fact, there is also adhesion which is not dependent on calcium. Of course, that is not regulated by cadherin, because cadherin mostly mediates calcium dependent cell adhesion, as I mentioned. But just you can remember the fact that the adhesion can also occur independent of the calcium, but that is through a different mechanism. That is not through this cadherins, because they are calcium dependent cell adhesion molecule. We call them CAM. CAM in this case means cell adhesion molecule. So how do you show that calcium is needed? I already told you, if you use some calcium chelating agent, then there will be shortage of calcium and therefore the cadherin will not function. Cadherin will not work. Now, in case of intermediate filaments, the terminology is like, for example, as I said, in desmosomes and hemidesmosomes, That is, we are now not talking of actin, we are now talking of the intermediate filaments. There also you will find some button-like points of intercellular contact, and they help in the anchoring sites for the rope-like intermediate filaments. But in this case, the structure is more or less the similar. If you take a look at the right hand side figure, so you can see again that there are some green components which are shown, which are having similarly a head portion and a tail portion. Okay, so the structure is more or less like the cadherin only, but the names are different. Here you will not call them cadherin. What do you call these proteins? Desmoglin, desmocholine. These are some of the proteins. And the tail portion, in the tail portion, you find certain proteins called like placoglobin. This is another protein. Desmoglin, desmocholine, these are cadherin family adhesion proteins. They are cytoplasmic tail. It binds placoglobin. Placoglobin is similar to gamma catenin. That is the protein that we mentioned or that we named as catenin. In case of cadherin, we will name those kind of proteins as placoglobin. And that placoglobin in turn binds to the desmoplakin. And that desmoplakin in turn communicates with the intermediate filaments. So that is how, the cell, any cell, it attaches to the intermediate filaments. So cell attaches with the extracellular matrix, cell attaches to the actin filaments, cell attaches to the intermediate filaments. The protein components, they are named differently. So you have to remember some of the names of the proteins or examples of proteins which are helping in this binding or anchorage. Dense cytoplasmic plaque composed of a complex of intracellular attachment proteins, placoglobin, desmoplakin, responsible for connecting the cytoskeleton to the transmembrane linker or adhesion proteins. Now here I have mentioned about one disease called pemphigus. This pemphigus is a fatal skin disease. What happens in this disease? Individual, they make antibodies against their own desmosomal cadherin proteins. Your body suddenly starts to produce antibody. Normally antibody is produced during infection. But what happens? In these patients, the body suddenly starts synthesizing antibody against your own protein. That is own desmosomal cadherin protein. And what is the result? Result is that the skin epithelial cells are destroyed. It is a skin disease. Skin epithelial cells are destroyed. And therefore there is severe blistering. And there is leakage of body fluid into the loosened epithelium, because your epithelial cells, they get loosened. And therefore all the vital body fluids, they are leaked out of the body, because there are leakage or there are blisters in the skin. But in this case only the desmosomal proteins, desmosomal cadherin proteins, they are defective. And due to their defect, this kind of skin disease, they occur. So what I mean to say is that how important these desmosomal cadherin proteins are. Intermediate filament proteins are also very important, not only for maintaining the structure, but also for, the physiological activities, normal physiology of any animal. Similarly, hemidesmosomes. So why hemidesmosomes? Because these are, hemi means partial. The distribution of the desmosomes and hemidesmosomes of the small intestine, like for instance keratin intermediate filament networks of the adjacent cell, they are connected to one another through desmosomes, and to the basal lamina through the hemidesmosomes. So hemidesmosomes or half desmosomes, instead of joining the adjacent epithelial cell membrane, they connect the basal surface of epithelial cells to the underlying basal lamina. So here also you will find several transmembrane linker proteins. Like these proteins are called the integrin protein, which bind to the extracellular matrix. So we will study about the integrin protein little bit when we will study the extracellular matrix. Then we will study in brief about the integrin proteins. But today we are not focusing on integrin proteins. So that is all about the structural components of the three different types of junctions. Now, before I end, I will just try to share maybe 3 or 4 more slides where we will classify the cadherins, and we will show how the cadherins, they help in cell signaling process. Now, what are the different categories of cadherin? E-cadherin, N-cadherin. In that way, we give different names to the cadherins. What is E-cadherin? E stands for the epithelial cells. N-cadherin, N stands for the nerve cells. So what does it mean? It means that the cadherin molecules are different in different tissues. The cadherin which you find in the epithelial cell and the cadherin which you find in the nerve cell, those are different. Of course, the E-cadherin which is present in the epithelial cells, it helps connect the cortical actin cytoskeleton of the cells, and they are held together. E-cadherin is also the first cadherin expressed during mammalian development, and it causes compaction, an important morphological change that occurs during the mouse embryo development. Like for instance, the formation of the blastomeres. During compaction, the loosely attached cells called the blastomeres, they become tightly packed and joined by intercellular junctions. So if you use antibody against E-cadherin, what will happen? E-cadherin will be destroyed and it will arrest the blastomere compaction. And therefore, these normal embryological processes, they will get hampered. So what does it mean? That means cadherin also helps in this kind of embryological phenomenon or the embryogenesis. So they not only play the structural role, but they also help in this kind of physiological mechanism. Now, when there is a embryo formation, then there is no demarcation of the cells. Like one cell which is destined to form the notochord, and another cell, let's say it is destined to form some other cell. Okay, initially they are all mixed up during in the embryonal stage, but later the cells need to get separated and they need to get sorted out, because all the similar cells will get sorted to give rise to a particular tissue, say a notochord, and some other cells will actually aggregate to give rise to a different tissue. So on what basis this kind of cells, they distinguish or they get sorted out and then they get aggregated? It is based on cadherins. So a cell which is having a particular type of cadherin, say a epithelial cell, it is having E-cadherin. What will happen? It will only get separated and then it will only associate with cells containing E-cadherin only. It will not associate with cells which contain N-cadherin. So all the cells which are having the E-cadherin, they will aggregate and they give rise to a particular tissue. All the cells having N-cadherin, they will separate. They get sorted and they get assembled to give rise to a particular tissue. So you can see how important these cadherin molecules are, because it is based on this cadherin that the cells will aggregate and they will try to associate, and they will give rise to a particular kind of tissue. So cadherins mediate cell-cell adhesion by a homophilic mechanism. So homophilic binding, that means that the molecules are of the same kind. It's the same kind of molecule, that is, in case of homophilic binding, you have that molecules on one cell, they bind to the other molecules of the same kind on adjacent cell. In heterophilic binding, the molecules on one cell bind to molecules of a different kind on adjacent cells. And in linker dependent binding, cell surface receptors on adjacent cells are linked to one another by secreted multivalent linker molecules. So if we diagrammatically represent these three mechanisms, then it will look like this: homophilic binding, heterophilic binding, and binding through intracellular linker molecule. And in the next figure, it has been shown that what I discussed as cadherin dependent cell sorting. That is, I just repeat once more that if you have a collection of cells, aggregation of different kinds of cells, then you will see that the cell has a tendency, or the cell has that property where it can sort out according to the type and the level of cadherin. So the red colored cells are having one type of cadherin, the blue colored cells are having a different kind of cadherin. So the blue colored cells will assemble together and the red colored cells will assemble together after they get sorted out, and then they give rise to appropriate tissues. So the cadherins not only help in structural component, not only act as structural component, but also helps in signaling role, or they also play a functional role. So with that, we just try to summarize what we have studied today, and then we will wind up. So basically, we discussed about the different types of junctions, the occluding junctions, the adherens junctions, and the communicating junctions. We discussed each of them in detail. What are their functions? What are the different structural components? What are the different proteins? How these proteins are organized? Okay, either in case of vertebrate, invertebrate, or whether in case of plant and animal, like in communicating junctions. Okay, and in case of adherens junction, how the two cells are attached, how a cell is attached to the actin filament or intermediate filament, also how the cell is attached to the extracellular matrix, and then what are the different physiological or the signaling role played by each of these components. So please go through the unit. It is in block 4, the first unit. We have talked about one part, that is cell junction. In my next session, maybe I will take up the second part, that is the extracellular matrix, and we will discuss the different structural components and the function of the extracellular matrix. So please go through the module, enjoy your learning, and get back to us in case of any problem or any confusion, any clarifications. We will be there to help you or assist you. So have a very, very good learning. Thank you all.